This is the Unit 3, 4, and 5 Anatomy Project poster video for the first target for Unit 3. I have compare and contrast skeletal muscle cells with typical animal cells. So let's take a closer look at the skeletal muscle cells and how they differ from typical animal cells. Right away, you'll notice that the skeletal muscles are huge in comparison to the long, cylindrical, multinucleated, meaning they contain multiple nuclei in a single cell. This allows them to produce lots of proteins for contractions, and these cells are packed with myofibrils, which are bundles of myofilaments made of actin and myosin, which are proteins essential for contraction. They're organized in a very specific way that creates striations, the light and dark bands we see under a microscope. Compare that to the standard animal cell, which is small, which usually has one nucleus and lacks this kind of organization. It performs more general functions like metabolism, growth, and repair. Skeletal muscle cells, on the other hand, are specialized for one main job: contraction. They have a sarcomella instead of a typical plasma membrane, and they contain a sarcoplasmic reticulum to regulate calcium ions, which are essential to innate contractions. This highly specialized structure allows skeletal muscles to carry out its specific function of movement. It's a great example of the theme structure determines function. Without this unique design, muscles can contract with force or precision when we need to walk, breathe, or even smile. The next target for Unit 3, Target 2, is sequence the steps of muscle contraction. Now let's look at a breakdown of the most incredible processes in the body, muscle contraction. It all starts with the neuromuscular junction, or the point where a motor neuron meets a muscle fiber. When a nerve impulse arrives at the axon terminal, it triggers the release of acetylcholine, a neurotransmitter, into the synaptic cleft. This chemical crosses the gap and binds to the receptors on the sacromella, the muscle fibers membrane. The binding opens up sodium ion channels. The sodium rushes to the muscle cell, creating an action potential. And once the signal is strong enough, known as the threshold stimulus, the sarcoplasmic reticulum releases stored calcium ions. The calcium binds to the proteins on the actin filaments, exposing building sites. Now, myosin heads, powered by the ATP, latch onto these sites and pull, causing sarcomere to shorten. As Z discs are pulled together, the entire myofibril contracts. This cycle repeats many times in a second, allowing for smooth, controlled movement. The key takeaway: this process is highly structured, relying on precise timing and coordination of different parts. Every piece of the synaptic vessel to the myofilaments is critical for function. Unit 3, Target 3. Identify the components of a sarcomere. The sarcomere is the functional unit of a muscle contraction. You can think of it as the smallest building block that allows the muscles to shorten and move. It exists between two Z discs and contains the thick myosin filaments and thin actin filaments. These filaments are arranged in highly organized way that gives skeletal muscles its striated appearance under a microscope. During contraction, myosin heads attach to the actin and pull the filaments towards the M line, which shortens the H zone and draws the Z disc closer. This shortening is what creates the force in movement. The A band stays the same length because it's the entire length of the myosin filament, but the I band and the H zone shrink. Every tiny shift at this microscopic level adds up to the visible muscle movement. The sarcomere structure is precisely what allows it to perform its function. It's built for repeated rapid contraction. It's like a perfect example of the body's design serving its purpose. Unit 3, Target 4, describe the organization of a muscle fiber. Muscles might seem like a solid chunk of tissue, but inside they're carefully organized at many levels. At the smallest level, we have the myofibrils, which contain repeating units called sarcomeres. The units are responsible for contraction. These myofibrils are packed into muscle fibers, which are long cylindrical cells surrounded by connective tissue called endomysium. These fibers are then grouped into fascicles, wrapped in perimysium. Finally, many fascicles form a whole muscle, which is surrounded by epimysium. This layered organization is just for structure. It plays a key role in function 2. It allows the muscles to contract efficiently, helps isolate and repair damage. It also provides pathways for nerves and blood vessels. At the end of each muscle, the connective tissue blends into tendons, which anchor muscles to bones or other tissues. This lets force be transmitted from contracting fibers all the way to the skeleton. Unit 3, Target 5. Impact of muscle disorders. Muscles do so much more than just help us move. While we usually think of muscles as a tool for walking, lifting, or exercising, their role in the body is far more complex and essential. The skeletal muscles are not only power movement, but also help us maintain posture and stabilize joints, keeping us upright, balanced, and steady every day. Muscles are important for maintaining homeostasis, especially body temperature. When you're cold, your skeletal muscles automatically contract rapidly in a process called shivering, which produces heat and helps keep your core temperature stable. But muscles go even deeper than that. Smooth muscles, which line the organs of the muscles, like the stomach and intestines, play a vital role in digestions. They perform peristalsis, which is a wave-like contractions that move food through the digestive system. Because muscles are involved in so many essential functions, it's no surprise when something goes wrong, the effects can be very serious. Some of the major muscle-related disorders are muscle dystrophy. It's a genetic disease where mutations interfere with the proteins that keep muscles strong. Over time, this causes muscles to weaken and break down. And then the next one is myasthenia gravis, is an autoimmune disorder where the body blocks nerve signals from reaching the muscles, leading to muscle weakness, especially in the face and eyes. Muscle strains and tears happen when muscle fibers are overstretched or torn. This leads to pain, swelling, and limited movement, and is often caused by overuse or sudden movement. Fibromyalgia causes chronic widespread muscle pain and fatigue. The exact cause is unclear, but it may be linked to how the brain processes pain signals. Rhabdomyolysis is a serious condition where muscle tissue breaks down rapidly, releasing a protein called myoglobin in the blood. This can damage the kidneys and cause darker colored urine and fatigue. Parkinson's disease is a brain disorder that affects movement. It causes muscle stiffness, tremors, and slowed motion due to the loss of dopamine producing neurons. Polio is a viral infection that attacks motor neurons in the spinal cord, often leading to muscle weakness or even permanent paralysis. Tetanus is caused by bacterial toxin that leads several uncontrollable muscle spasms. Even simple movements can become intensely painful as the muscle contract constantly. All of these conditions remind us how essential healthy muscles are, not just for movement, but for the basic functions that keep us alive and well. Unit 3, Understanding. My initial understanding at the beginning of the unit was surface level. I thought them as body parts that simply move things, and I associated with how strong or big someone looked, like the arms or the legs. I didn't know the actual names, layers, or types of muscles involved. So, I also didn't understand how different types of muscle fibers play different roles in performance, or how muscle fatigue works. What changed my understanding was the gross muscle anatomy notes. Learning about the names, location, and actions of muscles like the zygomaticus, the rectus abdominis, the deltoid, the pectoralis major, helped me realize just how specialized muscles are, not just for strength, but for expression, posture, and stability. For example, I never thought about how the buccinator helps hold food in place, or how the deltoid can move the arm in multiple directions depending on which fibers are used. Then also the muscle fatigue lab also helped me by actually doing the clothespin and the textbook challenges, which made everything feel more real. I felt how my muscles burned quicker during anaerobic, fast-paced challenges, and how it could last longer during the aerobic endurance challenges. It was clear that oxygen and ATP were important to performing. When they ran out, so did my energy. Feeling the burn was no longer something like I heard about. I understood it was lactic acid buildup from anaerobic respirations. The three ways my understanding has changed is I used to think all muscles did the same thing, just contracting to move body parts. Now I understand that muscles have specific structures and action depending on their role. For example, masseter is short and thick for forceful chewing, while the orbicularis oculi is a circular muscle just for blinking. And then before I didn't know there were different types of muscles. Now I know we have slow twitch muscles, which are red, aerobic, and fatigue resistant, and fast twitch, which are white, anaerobic, and fatigue prone. And the training that can change which type dominates. This explains why like sprinters and marathoners train differently and why they look and perform so differently than other athletes. I now understand why muscles get tired, and it's just because I use them too much. It's because ATP runs low, oxygen runs out, and lactic acid builds up during anaerobic respiration. The lab really helped me connect these ideas. For example, I noticed I could do the clothespin challenge for longer when I paced myself, but felt the burn much faster during the speed rounds, especially in my non-dominant hand. This unit's helped me move from surface level understanding to a deeper functional understanding of the structure, fiber type, and energy use. I now see muscular system as an incredibly organized down to the type of respiration cell use and the fiber types they contain. It's no longer just about who has the bigger muscles. It's about how they're used, trained, and fueled. Unit 4, Digestive System. Target 1. Describe the function of the digestive system and identify the organs responsible for carrying out these functions. The digestive system plays an important role in maintaining homeostasis. By breaking down the food we eat into molecules, the body can absorb and use for energy, growth, and repair. The system's main function includes ingestion, which is taking in food, and digestion, which is breaking down mechanically and chemically, absorption of nutrients, and elimination of waste. This process begins in the mouth, where mechanical digestion occurs through chewing, and enzymes like amylase start chemical digestion of carbohydrates. Food then travels down the esophagus to the stomach, where muscular contractions and enzymes like pepsin further digest proteins. Most absorption happens in the small intestines, which has a huge surface area thanks to the villi and microvilli, which are specialized structures that include nutrient absorption. The large intestine absorbs the water and minerals, preparing waste for the elimination, while the accessory organs such as the liver produce bile to emulsify fats. The pancreas releases enzymes like lipase, protease, and amylase, and the gallbladder stores bile. These organs work together to optimize digestion and nutrient uptake, showing how the structure of these organs allow them to carry out their functions efficiently. Altogether, the digestive system maintains the body's internal balance by supplying nutrients and removing waste. It's like a perfect example of homeostasis in action. Unit 4, Target 2. Describe the structure and function of the accessory organs of the digestive system. Each organ of the alimentary canal shares a basic four-layered structure: mucosa, submucosa, muscularis, and serosa. But each layer varies slightly depending on the organ function. The mucosa is the inherent-most layer that lines the lumen where digestion and absorption happens. It contains specialized cells that secrete enzymes and absorbs nutrients. Next is the submucosa, which houses blood vessels, nerves, lymphatics, which are essential for transporting absorbed nutrients and regulating digestive activity. The muscularis is made of smooth muscle arranged in two layers, circular and longitudinal, that contract rhythmically like waves called peristalsis. To push food through the tract. This layer is especially thick in the stomach, which needs powerful contractions to churn food into chyme. Finally, the serosa is a protective outer membrane that reduces friction as organs move. These layers adapt to the role of each organ. For example, the small intestine's mucosa has finger-like projections called villi to increase the surface area for absorption, while the esophagus has a tougher mucosa to protect against abrasion. This layer design shows how the structure of each alimentary organ allows it to perform its specific function effectively. And there are 4 parts of the accessory organs. The first one is the liver. It produces bile and to break down fats and detoxify harmful substances. The gallbladder stores and concentrates bile, releasing into the small intestine when needed. And then the pancreas produces digestive enzymes and bicarbonate to neutralize stomach acid, releasing them into the small intestine. And then the salivary glands produce saliva containing enzymes that start chemical digestion of carbohydrates. Unit 4, Target 3. Compare and contrast human digestive system versus animal system. The human digestive system is designed to handle many kinds of food. The humans are omnivores, which means we eat both plants and animals. Our digestive system needs to be flexible to process different types of food. We have a single-chambered stomach, so all the food we eat is digested in one place. Our teeth show what we eat. We have sharp teeth in the front to cut and tear food, and flat teeth in the back to grind it. Our intestines are medium in length, which help us digest both plant materials and meat as well. On the other hand, animals have many different kinds of digestive system depending on what they eat. Herbivores like cows and deer only eat plants. Plants have tough fibers that are hard to digest, so many herbivores have special stomachs with multiple chambers. For example, cows have 4 stomach chambers. In these chambers, tiny bacteria help break down the plant fibers through a process called fermentation. Cows even bring back their food to chew it again, calling chewing the cud to help digestion. Carnivores, such as lions, wolves, and only eat meat. Meat is easier to digest than plants. Carnivores have sharp teeth for tearing the flesh and shortened intestines because their food breaks down quickly. The stomachs are very acidic to help digest proteins and kill harmful bacteria. Omnivores like pigs and bears eat both plant and animal. So their digestive systems are a mix between herbivore and carnivores. They have one stomach, mixed types of teeth, and intestines that are medium in length, which are similar to humans. Looking at the teeth and intestine length tells us about an animal's diets. Herbivores have flat teeth for grinding and long intestines to absorb nutrients from plants. Carnivores have sharp teeth and short intestines to digest meat quickly. Omnivores have both kinds of teeth and medium-length intestines. These are the differences that show how animals have changed over time to match the food they eat. So while humans have a flexible digestive system that can handle many types of food, many animals have systems that are specifically made to digest only certain types of food well. This makes them very good at digesting what they usually eat, but less able to eat other kinds of food. Unit 4, Target 4. Analyze the actions of digestive enzymes. Identify where these enzymes are made and found throughout the digestive tract. Explain the relationship between an amylase and a specific substrate. Digestive systems are special proteins called biological catalysts that help speed up the chemical breakdown of food molecules. This process is important because it changes food into smaller parts that the body can absorb and use as nutrients. Each enzyme is very specific, which means it only works for one kind of food molecule, which shows how a key fits into only one block. For example, amylase targets carbohydrates like starch and breaks them down into simple sugars such as glucose. These sugars are easier for the body to absorb and use for energy. Proteus works on proteins, breaking them down into smaller units called amino acids, which are important for building muscle and repairing tissue. Lipase, it breaks down fats, also known as lipids, into fatty acids and glycerol, which the body uses for energy storage and to build cell membranes. These enzymes are produced and released in different parts of the digestive system to make sure digestion happens properly. Salivary amylase begins the digestion of the carbohydrates right in the mouth as we chew food. Once the food reaches the mouth, an amylase starts called pepsin, which is a type of protease, starts breaking down proteins in the acid environment created by stomach acid. The pancreas is a very important organ that produces many digestive enzymes, including amylase, protease, and lipase. These enzymes travel through the tubes called ducts and are released into the small intestines, where most digestion nutrient absorption happens. The small intestine also produces its own enzymes. The digestion process. These enzymes break down food molecules into smaller parts, simple sugars, amino acids, and fatty acids so they can pass through the walls of the small intestine and enter the bloodstream. This step is very important because it allows the body to absorb nutrients it needs for energy, growth, and repair. Because enzymes are so specific, they make digestion very efficient. They only cause the right chemical reaction, which prevents problems in the body. The careful control supports the body's balance, known as homeostasis, by making nutrients available when the body needs them. Without these enzymes working properly, digestion would be slow or incomplete, and the body would not get enough nutrients to stay healthy and function well. Unit 4, Understanding. At the start of this unit, my understanding of the digestion system was mostly basic. I knew food was broken down and nutrients were absorbed, but I thought digestion was simple, like a one-step process happening mostly in the stomach. I didn't fully understand the different layers of the alimentary canal. I didn't even know there was an alimentary canal, or how specific organs and enzymes contributed differently to digestion. Also, I hadn't considered how much the accessory organs like the liver, gallbladder, and pancreas beyond their basic names. What changed my understanding was studying the wall of the alimentary canal, learning that the digestive tract has 4 different layers: the mucosa, submucosa, muscularis, and serosa, with each unique types and functions helped me appreciate the complexity of how digestion and absorption happen physically and chemically. And then going through the detailed notes like the alimentary canal and the digestive system overview notes help me understand how different parts of the system specialize in different parts of digestion and absorption. For example, the stomach's muscular layers produce churning, the small intestine's villi increase surface area dramatically. And then I also learned that focusing on the salivary glands, liver, and gallbladder, and pancreas, and their secretion, enzymes, biles, and bicarbonate helped me see that digestion is a team effort, with the accessory organs providing crucial chemicals that help in breaking down fats, carbs, and proteins and protecting that digestive tract. How my understanding has changed is before I did not know that alimentary canal had full 4 layers with different tissues forming specific roles, from mucosa secreting enzymes and absorbing nutrients to muscularis contracting to move food. Another thing is I now understand that the liver produces bile to emulsify fats, the gallbladder stores and releases this bile, and pancreas releases enzymes like amylase, protease, and lipase, plus bicarbonate to neutralize stomach acid. These organs don't handle food directly, but are very important to chemical digestion, which I hadn't fully appreciated. And last but not least, the small intestines lining with villi and microvilli greatly increase surface area to maximize nutrient absorption. Also, I learned that different digestive enzymes are produced and act at specific locations. For example, salivary amylase starts carbohydrate digestion in the mouth, pepsin break down protein in the stomach, and pancreatic enzymes finish digestion in the small intestine. Unit 5, Blood, Immune System, and Circulatory System. Target 1, Structures of blood and its components. Blood is an important fluid that keeps our body working properly in balance, which is called homeostasis. Blood is made up of 4 main parts, with each of its has an important job. The first part is plasma, yellowish liquid that makes up about 55% of your blood. Plasma carries nutrients like sugar, amino acids from food, hormones that control body functions, and waste products that need to be removed from the body. Next, we have erythrocytes, or red blood cells. Their main job is to carry oxygen from the lungs to every cell in the body. They do this with a special protein called hemoglobin, which binds to oxygen tightly. Without oxygen, cells cannot make energy and survive. Then there are the leukocytes, or white blood cells. These are the defenders of the body's immune system. They fight bacteria, viruses, and other harmful invaders that cause disease. White blood cells are like soldiers protecting a city. And then lastly, we have the thrombocytes, or also called platelets, which help the body clot when you get a cut or an injury. They quickly gather at the wound and form a sticky plug to prevent too much blood from leaving the body. Together, these parts of blood deliver oxygen, nutrients, fight infections, and repair damages, helping the body stay healthy and balanced. Unit 5, Target 2, Steps of blood stoppage, or coagulation. When you get a cut or an injury, your body quickly works to stop the bleeding to protect you. This process is called homeostasis, or blood stoppage, and it happens in 3 important steps. First, the damaged blood vessels do something called vasospasm. This means the vessels tighten and narrow to slow down the flow of the blood near the injury site. This is like turning down a faucet to reduce the water flow. Second, the platelets rush to the injury site. This small cell fragments stick to the damaged area and to each other, creating a sticky platelet plug. Think of it like a sticky patch covering a hole, or like tape covering like a broken cut in something. Finally, the body strengthens the plug by creating a mesh made of protein called fibrin. This mesh weaves through the platelets and traps the blood cells, forming a strong, stable clot that seals the wound. This step-by-step process quickly stops bleeding, prevents the germs from entering the body, and is essential for keeping the body stable and healthy. So first, the blood vessels tighten, platelets rush to the injury site, then fibrin makes a weave, which forms into a scab to stop and seal the wound so it can heal, and this helps our body keep from any bad stuff entering our bloodstream. Unit 5, Target 3, specific versus non-specific immune defenses. Our immune system protects us from harmful invaders like bacteria and viruses in two main ways: non-specific defenses and specific defenses. Non-specific defenses act as the body's first line of defense and work against many different kinds of germs. For example, our skin acts like a barrier to stop invaders from entering. If germs get past the skin, sticky mucus in place like the nose and the lungs trap them. The acid in our stomach also kills many germs we swallow. Inside the body, white blood cells called phagocytes attack any invaders they find without needing to know exactly what it is. This is a fast general response that prevents infections early. The second defense is specific and targets particular germs. Special white blood cells called lymphocytes are involved, including B cells and T cells. B cells produce antibodies, proteins that attach to specific germs to mark them for disruptions by other immune cells. There are different types of B cells. Plasma cells make antibodies during infections, and a memory B cell that stays in our body after infection to help respond faster if the germ returns. T cells have several important roles. Helper T cells coordinate the immune response by activating other immune cells. Cytotoxic T cells kill body cells that have been infected by viruses or become abnormal, like cancer cells. Like B cells, memory T cells remain in the body to provide quick protection against future infections. Unit 5, Target 4, structures of the heart and blood flow. The heart is a strong muscular organ that acts as the body's pump, responsible for moving blood continuously throughout the network of blood vessels. It's roughly the size of a fist and is located in the chest between the lungs. The heart has 4 chambers arranged in 2 pairs. The upper chambers, called the aortas, a single is called an atrium, and the 2 lower chambers called the ventricles. The aorta receives blood coming into the heart, and then while the ventricles pump blood out of the heart. The blood that has delivered oxygen to the body and is low in oxygen returns to the heart and enters the right atrium. Through the large veins called superior and inferior vena cava from the right atrium, the blood flows through the tricuspid valve into the right ventricle. When the right ventricle contracts, it pumps blood into the lungs via the pulmonary arteries in the lungs. Blood passes through the tiny air sacs called alveoli, where it picks up oxygen and releases carbon dioxide as waste gas produced by cells. The now oxygen-rich blood returns to the heart to the pulmonary veins, enters the left atrium. From the left atrium, blood moves to the mitral valve into the left ventricle. The left ventricle has the thickest muscular wall because it needs to generate enough force to pump blood through the entire body. When it contracts, the blood is pushed into the largest artery in the body, the aorta, which distributes oxygen and nutrients to all organs and tissues throughout the small arteries and capillaries. Between each atrium and ventricle valve, including the tricuspid valve on the right side and the mitral valve on the left side, these valves function like a one-way doors, opening to allow blood to flow forward and closing tightly to prevent blood from flowing backwards. There are also valves at the exits of the ventricles, the pulmonary valve and the aortic valve. which prevent blood from returning to the heart after it has been pumped out. The heart pumping action is regulated by electrical signals that start in a small group of cells called the sinoatrial or SA node, often called the heart's natural pacemaker. These signals cause the aorta, the arteria, to contract first, pushing the blood into the ventricles. Then the signal passes to the AV node or atrioventricular node, which delays the impulse slightly before sending it to the ventricles, causing them to contract and pump blood out. This carefully coordinates structure and function of the heart, ensuring the blood flow efficiently in one direction, supplying the blood with oxygen and nutrients, while removing waste products. By maintaining continuous flow, the heart plays a critical role in keeping body's internal environmental state, also known as homeostasis. Unit 5, Target 5, Structure and Function of Blood Vessels. Blood vessels are tubes that carry blood all over the body, and their structure matches the job they do. Arteries take blood with oxygen away from the heart because the heart pumps blood with a lot of force. The arteries have thick muscular walls to handle the pressure and keep blood flowing smoothly. The veins bring blood without oxygen back to the blood. They have thinner walls because the pressure is lower, but they contain valves. These valves act like one-way doors, preventing the blood from flowing backwards, especially when blood moves against gravity. Between arteries and veins are tiny vessels called capillaries. These are so small that red blood cells pass through one at a time. Capillaries are where oxygen and nutrients leave the blood and enter body cells, and waste products move from cells into the blood to be removed. These specialized cells and blood vessels ensure blood flows efficiently, delivering what the body needs and removing waste, which helps maintain a healthy balance or homeostasis. Unit 5, Understanding. At the beginning of the unit, I had a very general understanding that the blood carried oxygen and helps fight infections. I knew the heart pumped blood, but I didn't know specific components of blood or how exactly blood clotting works. And regarding the immune system, I thought white blood cells just fight germs in a broad way. I didn't understand how different immune cells function or how body defends itself specifically and non-specifically. I changed my understanding to notes like the blood notes 1 and blood notes 2 about red blood cells, platelets, white blood cells, and plasma, the coagulation process, immune system defenses, and heart and blood vessel structure. And together, those help me really understand how these systems maintain homeostasis. I like studying those diagrams in the notes, especially in the innate immune system notes, help me understand how blood and immunity played hand in hand. So the ways my understanding changed is I thought the blood was mostly red blood cells carrying oxygen. Now it includes plasma, red blood cells or erythrocytes, white blood cells, leukocytes, platelets, thrombocytes, each with distinct functions like nutrient transport, immune defense, and blood clotting. For example, red blood cells contain hemoglobin to carry oxygen, and platelets form clots to prevent bleeding. I was unaware of that there were a multi-step process in clotting, like the vasospasm and platelet plug formation, coagulation, like the step-by-step response. And now I know that there's one which stops bleeding and prevents infection, which was new to me. Then before I thought all white blood cells did the same job. Now I understand there are two layers of immune defense, non-specific, like skin barriers, mucus, stomach acid, phagocytic white blood cells that protect against many invaders quickly and generally. Then specific defenses involving lymphocytes, B and T cells, where B cells produce antibodies targeting the specific pathogens, and T cells attack infected abnormal cells. This specificity and immune memory were concepts I hadn't fully understood before. Also, like, the heart was very, like, confusing to me. I knew that blood flow was started from the heart and came into the heart and went all over. I didn't understand that there was a difference between veins and like the arteries and veins. So like there was oxygenated and richly oxygenated and poorly oxygenated blood, and how there were many different like pathways, and that there were like 4 chambers of the heart. That was pretty interesting to me.